Hello friends, welcome to BMH Learning. We're going to talk about pseudogenes. So what is a pseudogene? Pseudogene is a DNA sequence that resembles a functional gene but has been mutated into an inactive form over the course of evolution. 2 points are highlighted in the given statement. First says, functional gene is mutated into an inactive form. That means pseudogenes are functionless or non-performing genes. We will reconsider this point again later in the lecture. The second point says it becomes non-functional during the course of evolution. So it's a type of evolutionary relic which indicates about the changing nature of genome. Now what's that which is actually making the pseudogene non-functional? Or in other words, what are the common defects in a pseudogene? The defects are in the form of coding and regulatory sequences. So that means a pseudogene will lack one of the upper mentioned sequences. And this sequence can be any one of the sequences shown in the diagram. The pseudogene may lack regulatory sequence such as promoter, which is needed for accurate transcription. It can be deficient in not having start codon, which is essential for initiating protein synthesis. And this becomes one of the reasons that a pseudogene doesn't make a protein product. The pseudogene can also be resulted because of a frame shift, a kind of genetic mutation which is caused by insertions or deletions of a number of nucleotides in a DNA sequence. Then you can also notice premature stop codon in a pseudogene. The presence of premature stop codon results in the formation of truncated proteins. In simple words, protein is not fully formed, hence it's not functional. And sometimes you are also going to observe the absence of introns in a pseudogene. Now let's discuss the most important concept about pseudogenes. Why do organisms keep pseudogenes? This question further points to many other questions, like why organisms maintain pseudogenes and pay a cost of energy. If maintenance of them is coming at the cost of energy, that means they are disadvantageous. If this is the case, why would not natural selection remove these costly DNA segments? If natural selection is not working, then what is the potential benefit to keep non-protein coding sequences? Although in the beginning of our discussion, I told you pseudogenes are non-functional, but let me tell you, they perform very important roles which make the asked questions quite interesting. Some pseudogenes appear to harbor the potential to regulate their protein coding cousins. It's normally assumed that they are silent relics, but many pseudogenes are transcribed into RNA, some of which are also activated in a tissue-specific pattern. Now there is a finding which suggests that pseudogene transcripts can be processed into short interfering RNAs that regulate coding genes through the RNA interference pathway. In yet another remarkable discovery, it has been shown that pseudogenes are capable of regulating tumor suppressors and oncogenes by acting as microRNA decoys. Now let's know different types of pseudogenes. Pseudogenes can be categorized into two forms. First is unprocessed pseudogenes, and the second one is processed pseudogenes. The unprocessed pseudogenes can also be subcategorized into two more types: the unitary pseudogenes and unduplicated pseudogenes. This was all. Thanks for watching.